Welcome back, students. Today we are going to see the anatomy of monocotyledonous stem, which is coming under the tissue system in dicots and monocots. Chapter 6, which is anatomy of flowering plants. So, as we have discussed about the anatomy of monocot and dicot root and dicotyledonous stem. So, accordingly, the anatomy of monocotyledonous stem in the ground plan shows epidermis, ground tissue, and vascular bundles. If you remember the transverse section and also the ground plan of the dicotyledonous stem, then we have seen that it was present as a ring structure, but here the vascular bundles are not arranged in a ring. That's why they are scattered all throughout the tissues. So this is the main reason why we will not find secondary growth in monocotyledonous stem as it was in the monocotyledonous root. So this is the ground plan and this is the transverse section of the monocot stem. So this transverse section of the monocot stem is prepared by cutting thin slices of the monocot stem, and after adding the appropriate amount of dye substance, it was observed under the compound microscope, and these were the structures. These are the arrangement of tissues which was found during the observation under the compound microscope. So let's see how these tissues are arranged in a monocotyledonous stem. So first, there is no epidermal hairs, but trichomes are absent. So sometimes epidermal hairs are present, but in few numbers. Then the first one is the outermost, which has having protective layer or protective function, is your epidermis. So epidermis is the outermost layer, and sometimes it bears epidermal hairs, but other times it won't bear. And as I said, cuticle is present in addition to the epidermis. Then next one is a hypodermis, and this hypodermis is composed of the sclerenchymatous tissues. So as it is evident that there is no intercellular spaces. So whenever you see that there is no intercellular spaces, then they are of sclerenchymatous cells. And importantly, the monocotyledonous stem is not differentiated into your endodermis, pericycle, and stick, which we are observing under the anatomy of monocot root, dicot root, and dicot stem. So here the monocot stem is not differentiated into endodermis, pericycle, and still, and also it doesn't have starch seeds, which was found in dicotyledonous stem of endodermis. Then what about the vascular bundles? These vascular bundles are not arranged in a ring, but they are scattered. The scattered arrangement of vascular bundle is the characteristic feature of the monocot. Then these vascular bundles, as they are scattered, so we can see here is a vascular bundle, here is one and another one. But the vascular bundles which are found towards the inner part, towards the central region, they are larger in size. So these vascular bundles are not arranged in ring but are scattered, and the vascular bundles are arranged by surrounded by sclerenchymatous term. So these are the sclerenchymatous having no intracellular spaces. So these intracellular spaces are sclerenchymatous 1, and this is the sclerenchymatous 1, which is known as a bundle sheath, and this is large conspicuous parenchymatous ground tissue. So this is your bundle sheath. This is made up of sclerenchymatous cells, and these the large ones, they are conspicuous and they are of parenchymatous nature. Then you have the vascular bundles. If you observe each vascular bundle, then these vascular bundles are conjoint and collateral. So in a vascular bundle, what we see, both xylem and phloem are present. So this is your phloem and these are your xylems. And as there is no vascular cambium, so it is a closed one. So closed type of vascular bundle is present, and conjoint one, xylem and phloem are present in a single vascular bundle. And as I have already mentioned, the peripheral or the vascular bundles which are found towards the peripheral region, they are of small one, or their size is relatively smaller than the centrally located vascular bundles. And here there is no pith, or pith is absent. So if we summarize the arrangement of tissues in a monocotyledonous stem, then we will see that the epidermis is the outermost protective layer, and it is additionally covered by a cuticle layer, and sometimes it bears epidermal hairs. Then hypodermis is of sclerenchymatous nature, and some of the cells contain your chloroplast, which are of parenchymatous nature, and that's why it is known as a chlorenchyma. Then the monocot stem, that is not differentiated into your endodermis, pericycle, and still or pith. So it doesn't have any starch sheath. Then vascular bundles are not arranged in rings, but they are scattered. Then vascular bundles are surrounded by, remember, vascular bundles are surrounded by a sclerenchymatous bundle sheaths, and towards the outer side they are surrounded by large conspicuous parenchymatous ground tissue. Then vascular bundles are conjoined and closed one. So conjoined means both xylem and phloem are present in a single bundle, and closed means there is no cambium in the vascular bundle, and that's the reason why the secondary growth is not shown by these monocotyledonous stems. And another feature is the vascular bundles which are found to the, or which are located towards the central region, they are of larger size than the vascular bundles which are found towards the periphery, but they are scattered all around the section. Then pith is absent. So remember the arrangement. So like that, what questions we can answer by understanding or studying the anatomy of monocotyledonous stem. First one is the description. Describe the anatomical features of a monocot stem. So this is one question which you can easily answer, and as we have discussed the anatomy of monocotyledonous stem and dicotyledonous stem. So another frequently asked question is differentiate between dicot stem and monocot stem. So you have to make a separate column for monocot stem and dicot stem and go on describing each point which they differ. Then as usual various short questions from here. So which of the vascular bundles are larger? So the centrally located vascular bundles are larger and the vascular bundles are not arranged in a ring-like structure but they are scattered. Then the various terms, what is protoxylem, what is your elder one. So as we have already discussed these short questions, so along with that prepare these questions and I hope you are getting proper and informative content from these videos. Keep learning, stay safe, take care. We'll meet in the next class.